All right, let's move on to lecture objective 3. This is where we distinguish between fast versus slow responses at the synapse. And so this is now where we're talking about what's happening when that neurotransmitter binds to that specific receptor, and how do we classify a response as being fast versus being slow. So this brings us to signal transduction. So signal transduction, the word transduction means conversion of energy or conversion of something from one form to the other. And so when we have that neurotransmitter binding onto this receptor, this is going to bring about a series of events in the cell, kind of like a complicated set of second messengers, right? So let's think about this on a simplistic level. Think about you telling someone to do something, they're going to tell someone else to do something else. And then there's this signal of events that are happening as this message gets passed on. And that's what the signal transduction really is. So when we bind that neurotransmitter to this receptor, we can have different types of messenger responses, different types of signal transduction responses that can be categorized as either fast or slow, depending on the type of receptor. So we've got these channel-linked receptors. These are also called ionotropic receptors. These specifically are fast receptors. And so when that neurotransmitter binds onto a fast receptor, it's simply going to open up that channel. We're going to have movement of an ion, and it's going to bring about an immediate response. Okay, we call this a fast receptor or an ionotropic receptor because the receptor is also doubling or also acting as a channel. Okay, and so this is why we say that it's fast. The change that's brought about in the membrane potential is going to be immediate or rapid because there's no passing of that signal the way I just explained. The channel is also the receptor, and so the response is going to happen as soon as that ligand or that neurotransmitter binds onto that, that receptor. And just to clarify what the word ligand here means, so we talked about this type of channel, we talked about ligand-gated channels, and we said that a ligand is anything that binds to a receptor and brings about a configurational change. And so in this case, the ligand here is the neurotransmitter, so we're using those terms interchangeably. So the neurotransmitter is what has to bind onto this receptor, bring about a conformational change, which opens up a channel and allows the flow of ions, which brings us our membrane potential change. Now, the second type of response here is called a slow response. And this is where we have several players involved, right? The message has to go through several different people before it can get to that cellular response. This is called a metabotropic receptor. And it's also coupled to a G protein. That's why it's also called a G protein coupled receptor. So here's our receptor. It's this transmembrane proteins, which is in the membrane here. Our neurotransmitter binds to this receptor, and the receptor is coupled or attached to a cytosolic facing protein, which we call a G protein. When that neurotransmitter binds to that receptor, because it's coupled to this G protein, this is where we're going to start to see the passage of that signal. So now the G protein is going to have a configurational change as well. We can see that the alpha subunit of this protein is going to dissociate off or detach from the rest of the protein. It's going to move downstream in the membrane. It's going to bind to another enzyme in the membrane, and it's either going to activate it or inhibit it, again, depending on the exact neurotransmitter that's involved. This can be a different response, either activating that enzyme or inhibiting that enzyme. When that happens, we generate what's called second messenger. And the second messenger are the players that are going to spread this message throughout the cell and actually bring about the cellular response. And this goes back to that example that I gave about you telling a story to someone else, right? Like, let's say you're passing a message or you're whispering, doing that whispering test where you whisper one thing to one person, they whisper it to someone else. And this is how that signal is passed. And so one, there are several players involved in the metabotropic or G protein signaling. Two, this takes time, right? It's going to have to go through several different stages. And so this is why we say that this is a slow acting or a slow type of signal transduction response. So the type of second messenger, again, can vary broadly depending on the actual cell, the actual neurotransmitter, but whatever that second messenger is, it's going to generate our secondary response, our cellular response, by either just opening up some other channel in the cell, we'll talk about some examples later on, or it will actually open up another ion channel in the membrane, okay? Now, just to kind of touch here, the second messenger can be a lot of different things. It's usually cyclic AMP. If you remember, we talked about cyclic AMP way back in biomolecules. It's one of those nucleotide derivatives, right? So adenosine monophosphate. And that is usually the second messenger, but it can be other things like calcium, etc. But basically, whatever that second messenger is, it is what is going to bring about our response, or the second messenger itself is going to open up another channel that will bring about the response. Another type of slow response or signal transduction is that we have a similar set of events initially. So we've got this G protein, the alpha subunit of this protein dissociates off, moves downstream in the membrane, but instead of activating or inhibiting another enzyme in the membrane, it's going to immediately activate or inhibit an ion channel that's in the membrane. And so this is going to be another type of slow response. This is called direct coupling. And so we kind of skip the second messenger and we go right to this ion channel as another way of bringing about this, this signaling mechanism. Okay, I'm going to pause here because that was a lot, and I'm going to see what questions you may have about fast responses versus slow responses, and just keeping those two ideas separated and how they are fast versus slow, why they're described as fast versus slow. Okay, so let's move on here. Actually, I see one question. Okay, so first question here is, are there certain responses that are always slow or always fast? So that's a good question. The receptors, one receptor versus another is always slow versus always fast. So for example, we'll see that the signal that controls muscle movement, skeletal muscle movement, is always a fast response, because again, our muscles are more responsive than our autonomic muscles. So for example, the type of synapse that controls the cardiac muscle is always slow. The type of synapse that controls skeletal muscle is always fast. Also, a neuron to neuron synapse is always fast. Again, I'll talk about the exact examples later on, but there are some type of signaling that's always fast versus always slow. Why are there two forms of responses? So that's another really good question, kind of tying back to what I just said. So think about it in terms of a structural and functional perspective. If remember, we're signaling here at the end of the day, we're sending signals. And so in order for me to send a signal from my brain to move my skeletal muscle, I want that to be very responsive, right? I don't want to have to wait for a delay, one messenger to tell another messenger, to pass the signal in order for me to move my hand, move my mouth, move my eyes, etc. This needs to be a fast, immediate response. So neuron to neuron synapses are always fast. Neuron to skeletal muscle synapses are always fast. Autonomic nervous system, so think about your glandular tissue, think about glandular secretions, think about your heart, the movement of your heart, cardiac muscle, think about smooth muscle, the GI system. These are not as responsive, and so these are always going to be slow responses, okay? And again, we'll clarify that when we talk about the autonomic nervous system, the skeletal muscle nervous system, the heart later on. But yes, we do have always fast responses versus always slow responses. They're not interchangeable. Okay, so really good questions, thank you. Let's keep going here. So let's talk about the graded potential and how that connects to what we're talking about now. So we've discussed this ligand-gated ion channel that's on the membrane of our adjacent cell. And so essentially the binding of our neurotransmitter is going to determine what type of graded potential is brought about on that adjacent cell. So I want you to kind of zoom out here for a little bit and remember that we're talking about one neuron signaling another neuron, right? From the brain to the muscle, all throughout our bodies. And so at the end of one synaptic communication is really the beginning of another synaptic communication. So essentially, when that axon terminal releases neurotransmitter, when it crosses the junction, it binds onto an adjacent neuron or adjacent cell. This is an opportunity for another action potential to be elicited on that adjacent neuron. And so this is where we begin to kind of talk about this in a cyclical way as far as generating another action potential. And so that neurotransmitter is really going to bring about graded potentials once again that could lead to another action potential on our postsynaptic neuron. And so I said all of that to say that if we open up a sodium or calcium channel, this is another graded potential. This is a depolarizing event. We call this an excitatory postsynaptic potential because it's a positive change in the membrane potential. So remember, sodium is positively charged, calcium is positively charged. Both of them have an ECDF that wants to move into the cell. And so whenever we open up these two types of channels, we're going to depolarize the membrane and on the adjacent cell, on the postsynaptic cell. And so we're going to generate a graded potential that's called an EPSP here on this second neuron. If we open up a potassium channel or a chloride channel, we're going to have an opposite effect. Because of the ECDF of potassium, we know it's in the opposite direction, it's out of the cell. And because chloride is negatively charged, if we open up either of these two ion channels, we're going to hyperpolarize the membrane or decrease the membrane potential. And so we can call this type of graded potential an inhibitory postsynaptic potential. So I really want you to make the connection here that this is kind of a full circle event. One action potential travels down one neuron, it's converted to chemical energy at the synapse, a neurotransmitter crosses the cleft, it binds to a receptor on the adjacent neuron or cell, and then depending on what type of ion channel opens up on that adjacent neuron, based on either a fast response or a slow response, then we can start to generate more graded potentials that could get us closer to threshold again, and we can fire another action potential on the postsynaptic, on the second neuron. And this is how action potentials move from neuron to neuron to neuron, being converted to chemical energy at the synapse, back to electrical energy at the axon, et cetera, all the way along that series of communication. All right, let's see, we've got some questions here. Excellent, so I'm getting a very good question here. Does the postsynaptic neuron act as a presynaptic cell when the process continues? And that is absolutely correct. And that is the connection that I want you to be making. This is not a fixed set of events, so don't be zoomed in on this one neuron, these one series of events. Kind of understand the bigger picture here. This is a series of communication between several different neurons, right? We've got billions of neurons in our nervous system. And so it's not just a one event and done, this is a continuous series of signaling events. So it's going from electrical energy to chemical energy at the synapse, back to electrical energy on another neuron, et cetera, or it could be converted to electrical energy on a muscle cell or a cardiac muscle cell or a glandular cell, if that is the specific cell that's being signaled. Okay, so excellent question here. And that's, again, the kind of lines I want you to be thinking along as far as really making the connection to the bigger picture here. Okay. So this is basically the postsynaptic potentials. I kind of mentioned this in a previous drawing that we did. And so postsynaptic potentials are really just graded potentials. They can be excitatory. When they are excitatory, we talked about the channels that they open up, either sodium or calcium. But what is the neurotransmitter that's released? It's usually glutamate. Glutamate is an excitatory neurotransmitter in our nervous system. Whereas if it were an inhibitory postsynaptic potential, this would open up a chloride or potassium channel. And so the neurotransmitter here is going to be different. It's going to be GABA, which is usually the inhibitory neurotransmitter of our nervous system. So again, I want you to be connecting that there's a different neurotransmitter that's released at a excitatory synapse than the neurotransmitter that we see at an inhibitory synapse. There's also a different type of channel that opens up at an excitatory synapse, right? It's going to be a sodium or calcium channel. And it can also be in the reverse direction. So I'm going to spend the last couple minutes here kind of clarifying this point here. So I'm going to jump back to this slide. So I talked about this as being excitatory if we open a sodium channel or if we open a calcium channel, but I want to make the connection here that this can be in the reverse. So if I close a sodium channel or close a calcium channel, that's going to be an IPSP. If I close a potassium channel or close a chloride channel, that's going to be an EPSP. So we can think about these as far as opening up the channel and having an EPSP if it's a positively charged ion like sodium or calcium. But if we close that channel, we're going to see the opposite type of graded potential. Okay, so just kind of thinking about this in the reverse as well.